welcome to our fourth lecture in the series. My name is Eden. I'm a data scientist in the MyOn lab, and today's lecture will talk about hypothesis testing, including multiple hypothesis corrections and some important probability concepts. So just as a quick overview of today's content, we'll start off by laying down some basic groundwork on hypothesis testing, just to orient ourselves and refresh some of the past knowledge we might have on this topic. So we'll go over the hypothesis testing framework and this important idea of the null distribution. And we'll talk about how we draw conclusions from observed data by specifying the significance level. Then a very important topic is the different types of errors that we can make in hypothesis testing, particularly in the case of multiple testing. So we'll talk about some common approaches to controlling these types of errors when we have multiple comparisons. So we'll cover the Sidak and Bonferroni corrections and the Benjamini-Hochberg correction for the false discovery rate. There's a lot of important concepts here that will be useful, not just in bioinformatics, but really any kind of statistical testing that you might be doing in the future. All right, so in scientific research, when we're trying to ascertain some kind of truth about the world, we start out with a hypothesis, which is a theory that we have, but we don't yet know if it's true. And then once we have obtained some real-world data, then we can use that to evaluate whether there's evidence to support the hypothesis. And this is what we mean by hypothesis testing. So hypotheses are going to be mathematically written as an equation or an inequality involving our variables of interest. And there's two forms of the hypothesis. There's the null hypothesis and the alternative. And the relationship between these two is that they are contradictory claims. So in other words, if the null is true, then the alternative must be false and vice versa. So for example, if we're going to test an equality relationship for a variable that we're studying, then the null hypothesis will assert that there is an absence of any differences. So we'd say the difference is equal to 0. And the alternative would assert that there is truly a difference, so the difference is not equal to 0. And an important point here is that in the hypothesis testing framework, the null hypothesis is designed to be the rejected claim, whereas the alternative will contain the researcher's expected findings. And we design it this way because most of the time in research, it's much more useful for us to disprove the opposite of what we expect than to try and prove something that we don't actually know that much about. So for instance, if I wanted to test the alternative hypothesis that there is some sort of difference between two populations, then really there are actually infinite possible alternative hypotheses I might have. So the true difference could be something small or it could be very large. We don't really have a great idea. So typically only the null hypothesis is actually going to be testable. So once we've defined the hypotheses, the next step is then to take the data that we've sampled and evaluate whether the hypothesis is true with a certain probability. And what we mean by that is that we won't be able to disprove our claim or our null hypothesis with absolute certainty, but we can use probabilities to essentially quantify how strongly our observed data supports the claim. And that's where we get p-values and confidence intervals, etc., which we'll discuss that in some more detail later. So in null hypothesis testing, we're essentially trying to see if our data disprove the null hypothesis. And we go about this by asking, okay, if the null hypothesis is in fact true, then how likely is it that we observed the data that we have? So to do this, we need a null distribution, which describes what the outcome should look like, assuming that the null is true. Specifically, we'll compute a test statistic from the sample data. So this could be a mean or a proportion or some kind of model parameter. And then the null distribution is going to describe the expected distribution of that sample statistic. So if we know the shape of this null distribution, then we can see where our observed statistic lies inside of it. And from there, we can calculate a probability of how likely it is for us to have observed the sample data under the null hypothesis. So just a brief clarification here about what we mean by probability distributions. So in statistics, there are some common named distributions that describe certain families of random processes or have important statistical applications. An example of this would be for count data. We often use the Poisson or negative binomial distributions to describe that sort of process. And then another example would be the binomial distribution, which describes the number of successes we would get from repeated trials with a fixed success probability. So it's obviously very nice and convenient when we have data that are well described by one of these known distributions. But that certainly won't always be the case. And in the next few slides after this, we'll talk about some ways that we can handle that. First, on the topic of formulating the null hypothesis, just to use the binomial distribution as an example here. Let's suppose we're collecting data on the number of free throws that an NBA star can make. So our null hypothesis is going to be that their accuracy is over 90%. So if this is our null hypothesis, then we know our null distribution is going to look like a binomial distribution. And then the parameter that will give this binomial distribution is a success probability of 0.9, as stated in our null hypothesis. So when it comes to defining the null distribution, a very foundational theorem I want to quickly mention is the central limit theorem, which essentially says that with sufficiently large sample size, the sample means will follow a normal distribution around the true population mean. So this theorem has very important implications for any statistical testing where we're taking the sample mean, because we can describe it with a normal distribution. All right, so after we've established our null distribution for our test statistic and we've collected our real-world data, then the next question is, how do we draw a conclusion about whether the null hypothesis is supported? So to do that, we use something called the alpha or the significance level, and this is essentially the probability at which you as the researcher would deem something to be sufficiently unlikely such that you would reject the null. So an alpha of 0.05 is kind of the standard level that people will often use. And that just means that if we observe an outcome with a probability of 0.05 or less under the null hypothesis, then that is sufficient evidence for us to reject the null. So if we consider this standard normal distribution on the right here, for example, if we obtained a sample statistic that lies in this middle region of high probability, then we'd say, okay, under the null hypothesis, it's pretty reasonable for us to observe this outcome, and we'd accept the null. But if the statistic we observe is very extreme, so maybe it's somewhere in these tails with sufficiently low probability, then we'd say, okay, under the null, it's very unlikely for us to have observed such an extreme statistic, and then we'd have the grounds to reject the null hypothesis. So we'll talk more about types of testing errors later, but the alpha level is actually equivalent to the type 1 error rate. So that means if the null hypothesis is indeed true, our tests would lead us to incorrectly reject the null hypothesis around alpha percent of the time. So if your alpha is 0.05, 5% of the time. So that's just something to kind of keep in mind, that increasing the significance level is equivalent to increasing the chances of committing type 1 error. And then one final important thing I want to reinforce here is that the only hypothesis we're testing in this framework is the null hypothesis. So in the end, our evidence is either going to support the null, in which case we accept it, or it will fail to support the null, in which case we reject it. And rejecting the null hypothesis simply implies that the observed data are very unlikely if the null hypothesis were to be true. So we're really not drawing any direct conclusions about the alternative hypothesis here. Okay, so just to get us started, we'll start with a simple example of hypothesis testing in a coin toss scenario. So suppose we have a coin and we're interested in finding out if it is fair or not by running an experiment with n tosses. So in this case, the null hypothesis is the probability of heads and tails being 0.5. So since a coin toss has two outcomes with a fixed probability of success, the number of successes we're going to get that's described by the binomial distribution, which I defined before. So the binomial distribution, it has this parameter p, which is the probability of success, and also the parameter n, which is the number of events that are occurring. And then we can calculate the probability of observing up to k out of k successes out of the n events through this summation here. So this comes from how the binomial distribution is mathematically defined. So now let's say we've tossed the coin 7 times and we recorded this data where we observed 2 tails out of and 5 heads. We can then use the formula from the last slide to precisely calculate what's the probability of observing this few tails, so either 0, 1, or 2 tails, if the coin is actually fair. So I've plotted the null binomial distribution here, which you'll notice is a discrete distribution, and our observed data lie at this green dashed line. So we can kind of see by eye already that it's not super unlikely for us to observe 2 or fewer tails under the null. And when we calculate the probability, it's around 0.23. So this is the p-value for our test. So if our alpha level was 0.05, then we would say we do not have sufficient evidence from this experiment to reject the null hypothesis. So now we know about how hypothesis testing is done in theory, but in practice, oftentimes the distribution of our data is unknown, or we don't have a closed mathematical form to describe it. So in the previous example of the coin toss, we were able to use the binomial distribution formula to directly calculate the probability under the null. But a lot of times we don't have that knowledge or an equation to do that precisely. So in those cases, a very useful alternative is to instead computationally simulate the event that we're studying to try and gain some insights into the outcome distribution. So the main advantage of this is that you don't need to have any prior knowledge about the outcome distribution, but in order to capture it sufficiently, it will require many repeated simulations, and especially if we're trying to capture a pretty complex distribution, this can get computationally expensive. So that's one limitation of this approach that you might want to consider. As an example in pseudocode here, suppose we wanted to simulate the outcome of that previous coin toss experiment. So we would do this if we really had no idea what distribution the number of successes is supposed to follow. So to simulate that process, we would perform 7 random draws and then count how many heads or tails we get each time out of the 7. And we would repeat this entire process of 7 draws n times. And then we can estimate the probability by seeing what proportion of those simulated experiments have 2 or fewer tails. And one thing to note about these simulations is that although we aren't making any assumptions about the outcome distribution, we are making plenty of assumptions about where the data are coming from. And those are potentially incorrect. So you want to make sure if you're doing this simulation testing that you're drawing from distributions that are most appropriate for the population being studied. Okay, so now suppose we really don't know that much at all about where the data are coming from, which is often true. So we have no idea about the data generation mechanism. Then another alternative for hypothesis testing that we can do here is to use permutation, also called shuffle testing. And this is a way you can simulate the outcome distribution without needing to generate your own random draws. Instead, you take your observed data and just randomly shuffle it, and you use these shuffled data sets as your simulated draws, from which you can then compute the test statistic and estimate the null distribution as we've done before. So for example, suppose we want to estimate if there is a difference in means between two populations. So this would imply we want to compute a t-statistic. So we're going to take the data from both populations that we have, and we'll generate n random permutations of that data. So we'll treat each of those permutations as kind of its own data set. And for each of those, we'll compute a t-statistic. And then afterwards, we'll have n t-statistics that allow us to estimate the null distribution. And as we did before with our known null distributions, we can see where our observed t-statistic lies in relation to the simulated statistics and calculate a p-value estimate from there. So something to note is that for n permutations or iterations, the p-value can only be as small as 1 over n. So basically, the more permutations you're able to do, the more accurately you can estimate the p-value. But of course, this would be at the cost of computational resources, especially if your data set is very large. This can get expensive very quickly. So there's a bit of a trade-off there. Okay, so now we'll move on to a slightly more biologically relevant example of hypothesis testing, which is testing for differential gene expression. So the topic of differential gene expression is an important one, and it'll get its own lecture next week. But for today, we're just going to discuss some basic concepts about hypothesis testing for differential gene expression. So suppose we've collected gene expression data from two types of samples, mutant and control samples. And we've done this under two different experimental conditions. We'll call them Lab A and Lab B. So in total, we have 6 samples. So 3 mutants, 3 controls. We've tested them under these 2 different conditions. So we have a total of 4 unique types of experimental conditions here. And then we've measured gene counts under each of these conditions. So our data might look something like the matrix here on the right. Our objective then is to test for differentially expressed genes between the mutant and the control samples. So these are genes that are expressed at different levels in the 2 sample types. And since we have this extra variable here, which is the lab condition, one way we can go about trying to test for differential gene expression is to fit a mathematical model for each gene that relates the gene's level of expression to these experimental variables. So one of them would be mutant versus control status. The other one might be Lab A versus Lab B. And this type of gene-wise modeling approach is what both DESeq and LIMA do to estimate change in gene expression between experimental conditions. So yeah, DESeq and LIMA, these are 2 widely used CG algorithms, which we'll talk more about next week. So suppose we have our data, and let's just pick a particular gene and plot the counts of that gene over the 4 unique types of experimental conditions that we have. You might get something that looks kind of like this very simplistic plot here. but you can tell the green control samples have higher expression than the red mutant samples. And then we can also see that lab B is having some effect that lowers the expression of the gene compared to lab A. So if we try to put this into a mathematical expression, let's have Y as the modeled outcome, the gene expression level. And let's suppose we set kind of the reference or baseline level to be the lab A control group. So we're going to give this an intercept term here that's fixed. Then the effect of lab B relative to lab A, we'll model that with this coefficient, as beta coefficient in front of an indicator variable that is equal to 1 if the sample is in lab B and 0 otherwise. And likewise, we can model the difference between the mutant and the control samples with a second term, so another coefficient and a second indicator variable that is equal to 1 if the sample is a mutant and 0 otherwise. So what we've done here is we've modeled a linear relationship between the gene's expression and the experimental variables, which are our input data. So it's a linear relationship, so you can see there's a linear combination of the input data. So this whole concept of linear modeling, it's very widely used in many different types of applications. So basically any model where the outcome variable y is dependent on some linear combination of explanatory variables x is considered a generalized linear model or a GLM. And in many cases, y is going to be dependent on the linear combination through what's called a link function, which essentially transforms the y into a different scale that is then linearly dependent on x. So for gene counts, we often use the log link. So if we look at kind of this notation here, it's the log counts that are linearly dependent on the explanatory variables here. So for gene I, we would model the log of its counts as a sum over these experimental parameters 1 through j. And based on this formula, each of these beta coefficients we interpret as the log fold change for gene I across condition j. So in a categorical case, if if xj represents some categorical variable, if it's equal to 1, then the log counts will increase by exactly beta ij. And what this means for the counts themselves is that they will multiply by a factor of beta ij. So that's what we call it, the log fold change. For RNA-seq count data specifically, it has this natural property of being over-dispersed, which means the variance of the counts tends to be greater than the mean. So the statistical distribution that a lot of software will use to model the gene counts is the negative binomial distribution, which has this dispersion parameter that captures that characteristic of RNA-seq data. So in our linear model, the beta coefficient or the log fold change is what we're going to use to quantify whether the gene is differentially expressed. So if beta is 0, then the condition has no effect on the gene expression level. So this is our null hypothesis. And you can imagine if it's positive, we're going to that indicates higher expression across the condition. If it's a negative value, it indicates lower expression. And when it comes to determining the null distribution of the betas, the precise details of that distribution, it's going to depend on how we're calculating the estimate for the beta coefficients from the data. It'll also depend on the noise that's inherent in the data. But in general, after we've successfully, have these estimators for beta and the standard errors, we usually use something like the z statistic or the t statistic. And this is what DESeq does. So they use the z statistic. And we would expect these statistics to follow the standard normal distribution if it's a z-score, or the t-distribution if it's a t-score. And something you'll notice about these statistics is that they're inversely related to the standard error. So basically, if we're able to decrease our standard error estimate, either by increasing the sample size or applying some shrinkage methods, which is what a lot of different software for differential gene expression will do, that will result in a more powerful test, which means we'll be more likely to detect a true effect. So just to return to our example from before, suppose we calculate this estimate for the beta coefficient of mutant versus control samples using maximum likelihood estimation. And we also have this estimate for the standard error of 2.6. Under the null hypothesis, which assumes that the log fold change is 0, we would expect the Emily over the standard error to follow this standard normal distribution. And this property of Emily comes from the central limit theorem. So just like we've done before, we would take our observed statistic, see where it lies in the null distribution, and then calculate a p-value for it to determine if there's evidence that the log fold change is 0. And in this case, we would say we do not have sufficient evidence to reject that null hypothesis. So when we're doing genomic studies, more often than not, we're going to need to be conducting many hypothesis tests at the same time. So far we've only talked really about single hypothesis testing, but especially in the field of bioinformatics, most of the time you'll probably be doing multiple hypothesis tests. So for example, with differential gene expression, if we've collected RNA-seq data from many genes, like over 20,000 genes, we're probably going to test almost all of them for differential gene expression. But there are some issues that arise from multiple testing. So to illustrate this, say we've performed 100 correlation tests between genes, and we want an alpha level of 0.05. So just by random chance, even if the null hypothesis was in fact true, we would find that roughly 5% of the genes we test are significantly correlated. So the more tests that you do, the more likely it is that you'll incorrectly reject the null at least once. You can imagine if we were testing 20,000 genes instead of 100, even if none of them are actually significant, we would probably still find that several hundred of them have a p-value less than 0.05. So that's a lot of genes, and if you're trying to isolate some meaningful genes to investigate experimentally, that's not very helpful information for you. So this is the issue that we're going to try to correct with our multiple comparisons corrections. So before we get into the types of corrections, we first want to define the types of errors that we're aiming to control for in hypothesis testing. So when we have sufficient evidence to reject the null, we call this a positive finding because it's in favor of our expected results. A negative finding would be the opposite, where we fail to reject the null hypothesis. So a type 1 error rate, which I mentioned before, is also called a false positive. And this occurs when the null is actually true, but we find evidence to reject it. And this error rate, it's determined by the significance level or the alpha level that we set. In the case of multiple testing, the corrections we have are going to aim to minimize this type of error. So another definition is the family-wise error rate. And this is the probability of making at least one type 1 error in a series of tests. So this is the specific measure that the Sidak and Bonferroni corrections aim to control. So those are the two corrections we'll be discussing next. And then the other type of error, which is not as important for today, but is still good to know about, is the type 2 error, or a false negative finding. So this occurs if the null is actually false, but we don't find the evidence to reject it. And this error rate is typically controlled by the sample size. So a larger sample size offers more power and then less chance of committing a type 2 error. All right, so we'll start off with the SIDAC correction. So this is a method to control the family-wise error rate at some desired alpha level. And it's not used as commonly as the Bonferroni, but the Bonferroni is actually an approximation of the SIDAC correction, which is why I'll introduce the SIDAC first. But how it works is for n comparisons, the SIDAC correction computes this new adjusted alpha level, which is given by 1 minus our desired alpha level raised to the 1 over n. So then after we have this adjusted threshold, we'll reject all the hypotheses, all the null hypotheses for p less than this new adjusted threshold. And by doing this, the derivation proves that we control at exactly the desired alpha level. But an important note here is that the derivation does assume all the tests are independent. So if there is any way that the tests are dependent, then this control over the family-wise error rate will deviate a bit from our desired alpha level. So if we do a Taylor expansion of this formula for the SIDAC correction, we get an approximation that will look something like this. And that first term here we see in this equation is what the Bonferroni correction uses. So yeah, the Bonferroni correction is the first order Taylor approximation of the SIDAC correction, and it has a much more simple form, and it is going to be at least as conservative as the SIDAC correction. So in other words, the Bonferroni p-value cutoff will always be less than or equal to the SIDAC cutoff. So for n comparisons, we just simply adjust our alpha by a factor of n. So to illustrate how this is controlling the family-wise error rate, just as an example, suppose we conducted 10 comparisons and we have a desired alpha of 0.05. The adjusted p-value, Bonferroni p that we would compute would then be 0.005. And so in this table here, I'm calculating the probability of committing a family-wise error. So this is just the probability that we have at least one finding under the null hypothesis, so one false positive. And that is equal to 1 minus the probability that we always fail to reject the null, which for n independent tests is equal to 1 minus the alpha raised to the n power. So without any correction, if we were just to use this cutoff of 0.05, we would see that our family-wise error rate is 0.4, which is a really high probability. On the other hand, if we were to apply the Bonferroni correction, so instead we're using this cutoff of 0.005, then our family-wise error rate is instead 0.049, which is under our original desired alpha of 0.05. So this is how the Bonferroni correction successfully controls that family-wise error rate. And an important consideration with both the SIDAC correction and the Bonferroni correction, though, is that when the number of tests is very large, the p-value cutoff becomes very, very small. And in those cases, these corrections are going to give you very conservative results. So for large numbers of comparisons, generally we'll consider some other approaches to controlling these type 1 errors. So that's where the false discovery rate, or FDR, comes in. So FDR is just the proportion of all the positive findings that are false. So essentially, if we say an FDR of 0.1, that would mean we're willing to accept that up to 10% of our significant results are incorrect. So this is especially useful if we're doing a lot of tests and we're looking to select some group of maybe gene candidates out of a whole bunch of genes. And kind of just an important distinction is that the false discovery rate and the significance level or the alpha level, they have quite different meanings. So if I were to present some gene candidates to you and say, okay, about 10% of these significant genes I found are actually not significant, you would still find most of those genes to be useful and meaningful for downstream analysis. But instead, if I were to say, actually 10% of all of the genes I tested are false positives, then you might not have a very strong degree of trust in these gene candidates that I'm presenting to you. So yeah, the false discovery rate and the alpha level, these are two different concepts. So to control the FTR, the Benjamini-Hochberg method is probably the most popular type of correction. And how it works is you'll first rank all of the p-values in ascending order, and then you'll find the largest p-value that is less than or equal to the desired FTR times the rank out of the total number of tests. And you take this p-value as your new significance threshold. Then rejecting all the hypotheses with p less than this cutoff, that will keep our false discovery rate less than or equal to that pre-specified level. So here it's denoted as alpha again. So we'll run through a quick example of how this correction is applied. So suppose we've conducted 5 tests and obtained these p-values, and we have a desired false discovery rate of 0.1. So generally this is kind of the highest like safe false discovery rate that you might want to use. So the first step will be to rank each of the p-values in ascending order. So after we've done that, then we can compute these BH critical values, which is just the desired FTR times the rank over the total number of tests. And then we're going to look for where we find the largest p-value that is no larger than that corresponding critical value. So that occurred in this second test here. And then all of the hypotheses with the p-values larger than that, we will say we failed to reject the null in those cases. So in some statistical packages, if you're using some software to do this, they might do this correction by computing a Benjamini-Hochberg adjusted p-value. So there, instead, they multiply the p-value by the total number of tests over the rank, and then apply the FGR level as the cutoff. So that approach is completely equivalent to what we just did. It's just a simple rearrangement of the inequality here. But in the notebook demo, that is the type of correction, or that is the calculation that we'll do for the BH correction. All right, so just to summarize some key points from today, we started out with the null hypothesis testing framework, where we use a null distribution to calculate the probability of an observed outcome. And this is how we get a p-value that we then use to decide whether we have evidence supporting the null assumptions. And then we talked about how in the cases of unknown or non-closed form distributions, we can use simulation testing to estimate the null distribution. Or if we don't have much knowledge about the data generation mechanism, we can do permutation testing from our observed data. Then another concept we discussed briefly is generalized linear models, which are commonly used to test assumptions about more complex relationships between an outcome and explanatory variables. And hypothesis testing for these GLMs will generally be about the coefficients in the model. And then finally, for performing multiple comparisons, we have the Monfroni correction to control the family-wise error rate. and the Benjamini-Hopper method to control the false discovery rate. I want to acknowledge Dr. Mayan for administering and kind of supporting this class series, and specifically for reviewing the content that you guys saw today, as well as Dr. Alex Lackman, who helps a lot with these materials today, and the rest of the Mayan lab members for their support in these efforts. So thank you, everyone.